Good morning, everyone, or afternoon, really, depending on the which part of the world you're joining us from. Like Ben said, I'm Ellie Juarez, and I'm the market segment manager for oncology here at Oxford Nanopore. Thank you all for being here with us. And today, I'll be talking very briefly about how nanopore sequencing can enable you to gain ultra-rich multi-omic data for your cancer research studies. And then I'll be passing it on to really the star of the show, Dr. Daniel Kem from UC Santa Cruz, who will go on to detail how his laboratory is leveraging nanopore sequencing for RNA liquid biopsies. So really just to set the stage here for everybody, Oxford Nanopore is a new generation of DNA and RNA sequencing technology, and really the only technology that offers real-time analysis for rapid insights in a fully scalable format from our MinION, which is the size of a cell phone, um, to population scale instruments in our Promethean line. Our tech can analyze DNA or RNA and sequence any length, both short to ultra long read, really making ONT an any length read technology. And all of this is enabled by a tiny nanopore embedded in a membrane. And then the DNA or RNA passes through this pore and emits a unique electrical signal, and then you can read this. And so these squiggles or electrical pulses you can see in this video here are just slightly different for an A versus a T versus a G, and importantly, slightly different for a C versus a methylated C. And so these unique and biological properties of the nanopore sequencing makes ONT a comprehensive platform for multi-omic studies. And this is because you can not only sequence the different types of nucleic acids at any length, but you can also assess their epigenetic modifications like M6A for RNA or 5-HMC for DNA. And this allows you really to get kind of a holistic view of the genome and the transcriptome and the epigenome, and all on a single platform. And I think this really comprehensive view is really crucial in the characterization of cancer, for which studies in the past of 5-10 years have shown that this multi-omic and comprehensive view of the genome is really, really crucial. At the end of the day, cancer is a disease of the whole genome, which is characterized by both those genetic and epigenetic changes. And then these changes drive aberrant transcription, protein expression, and ultimately these disruptive processes kind of serve as the mechanism for both the initiation of cancer or oncogenesis, a mechanism often for evasion of cancer treatment, and then unfortunately also relapse. And so being able to comprehensively view and probe both single nucleotide variants with phasing or fusion transcripts or gene expression on a single platform, we really believe will be crucial for the community at large to continue to do important studies to advance the oncology field forward, important studies like the work Dr. Kim's going to speak about today. So really with all of that in mind, and sort of as we look to the future of oncology research and clinical applications, the Holy Grail will not only be able to layer these multi-omic data, but also to be able to do so in a minimally invasive way for cancer early detection or monitoring. In 2022, Kutzman et al. Showed how to use ONT sequin and how you can leverage it for low-pass whole genome sequencing. And this allows for real-time results that, when coupled with ONT's fast sample preparation technology, allowed the researchers to get methylation-based tumor classifications in less than 3 hours. They also showed they could determine cell of origin using fragmentomic analysis on the ONT platform, and then further leverage that low-pass whole genome sequencing data for assessing copy number variation and even tumor fraction. And so the final layer of this full multi-omic study that they were conducting was the ability to leverage direct cell-free DNA sequencing for DNA methylation signatures. And this direct sequencing removed the need to use this degrading bisulfite or GC bias-based enzymatic conversion methodologies, and really ensuring that you don't waste a precious sample and often limited sample that you get from liquid biopsies. And this ultimately makes for more complex and informative sequencing libraries. I think Dr. Kim today will show us that direct RNA sequencing for liquid biopsy can also enable complete isoform mapping and transcript quantification. And importantly, I remind you that using ONT, you can also assess directly RNA-based modifications, which can also have been shown to be dysregulated in cancer. So as we look to the future of cancer and early detection and enabling researchers to tackle the unique challenges across the complete cancer journey continuum, we believe that by leveraging nanopore sequencing on a single platform, you can unlock multi-omic view of your cancer samples. And so while ONT can really be leveraged for an array of applications like hereditary cancer and hematological malignancies, Dr. Kim's talk will focus on leveraging ONT for probing epigenetic modifications and using long-read RNA sequencing to characterize the transcriptome in cancer. So without further ado, it is my pleasure to introduce Dr. Daniel Kim, who's an assistant professor at UC Santa Cruz. So, Dr. Kim, take it away.
Great. Thanks so much, Ellie. And thank you, Ben. Thanks, Seek Answers and Oxford Nanopore Technologies for this great opportunity. And thank you all for joining from wherever you are around the world. So I'm really excited to tell you about some of the work that my lab has been doing to advance some RNA-based liquid biopsy technology using the nanopore sequencing technology that Ellie kindly introduced for us. And so I'll be telling you about work that we've been doing over the past few years to really enable this platform to diagnose cancer at the earliest stages. And so, as many of you know, cells continuously secrete information throughout the body via RNA that's secreted in these extracellular vesicles. So if you think about all of the tissues and organs in our bodies, and you think about all the cells that comprise these different tissues and organs, it's estimated that there are about 37 trillion cells within the human body. These cells are just continuously sending out information about their state, whether they're healthy, whether they're diseased, into the extracellular matrix. And so if you look at these extracellular vesicles, or EVs, some of which may be exosomes or microvesicles, these are nanometer-sized particles that protect the RNA from degradation. So typically, if you think about what's called an exosome, they could be anywhere from 30 nanometers in diameter to about 150 nanometers in diameter. So these are, extremely small, again, nanometer-sized vesicles that, again, will protect any RNA that's secreted into, for example, the blood from degrading through these RNases that are present in the blood. And the great thing is, if you were to take a milliliter of blood from anyone here that's watching this webinar, that there would be billions of these EVs that are present in a single milliliter of blood. And so you can imagine, even if these EVs had only a single RNA molecule, you're talking about billions of RNAs that you can really profile using Oxford Nanopore Technologies, different sequencing platforms, or various other approaches, where you're able to identify signals that are coming from throughout the body. And so it really gives you this very dynamic and systemic information about what's happening within our organs and tissues. And so this is a really, I think, exciting area of research where now there's a lot of people focusing on extracellular vesicles and especially the contents of these EVs to be able to identify what's happening inside the body. And so if we think about kind of the RNA landscape of a given genome, for example. So within our human genomes, we know that we have about 3 billion base pairs that comprise the human genome. And what's been really quite interesting is that if you look at the output of the genome, most of the genome is actually making RNA. So it's thought that about 75% of these 3 billion base pairs is being transcribed into RNA. That may be kind of a conservative estimate. And if you think about it, when you look at all of the sequences in the human genome, only 1 to 2% of these 3 billion base pairs encodes for RNAs that make proteins, these protein coding messenger RNAs. And so the vast majority of the RNA that's being transcribed in the human genome is not making proteins, is not translated. It's actually non-coding. And so they exist as RNA. Some may be functional, some may not be functional. But the important thing is that we're really looking at this entire landscape. We want to leverage this landscape to be able to detect and understand disease in vivo. And if you think about kind of some of the ways that people have been trying to do this, as we heard from Ellie's introduction, there are these new technologies called liquid biopsies that people have developed to enable this in vivo profiling of what's happening in these very complex diseases such as cancer. And so liquid biopsies have really been transformative because they enable this non-invasive or minimally invasive way to sample different types of biofluids in the body. So our favorite biofluid is blood right now. So we're looking at the liquid fraction of the blood, the plasma, and really sampling what's happening within the blood and all of these signals, again, that are being released from these different cell types that comprise tissues and organs, especially really being able to get this, again, systemic molecular information, including from the brain. And so these extracellular vesicles, such as exosomes, have been demonstrated to cross the blood-brain barrier. So you're even able to get information about different types of cancers in the brain or even neurodegenerative diseases, which my lab has also been studying. And so really, again, the great thing about this approach of using a liquid biopsy is it really enables this continuous monitoring of these very dynamic and evolving disease states. And so we know that cancer is a moving target, right? As cancer continues to progress and develop, it will evolve genetically, epigenetically, at the transcriptomic level. And so really being able to monitor this in a continuous and dynamic manner is really transformative for understanding how the disease progresses, how disease may respond to different types of treatments. So you can imagine if you were to treat, let's say, a patient with immunotherapy or some small molecule drug, how is the patient responding to that particular therapy? And so this RNA-based liquid biopsy approach that we're developing really enables us to understand how patients are responding to these different types of, for example, drugs or treatments. And the reason why we really wanted to pursue this RNA-based approach is because existing methods may miss very early stages of disease. And so there have been some studies that have been published where people have looked at, for example, DNA or proteins that are circulating in the blood to be able to identify cancer. And especially for early stage disease, oftentimes there's not a ton of signal there. And so we wanted to identify alternative orthogonal approaches that would enable early detection of cancer and other diseases. And so if we think about, cancer, this is a tremendous global burden. We know that there are estimates of about 20 million people who were diagnosed with cancer in 2022 alone. And again, this is, a global problem. And unfortunately, if you look at kind of the number of people that have died from cancer, in 2022 alone, it's estimated that about 10 million people died from cancer. And the reason why, most people die from cancer is because the cancer has spread throughout the body, it's metastasized. And unfortunately, many of these cancers are detected at very late stages when the cancer has already spread and when there are not a lot of options for these patients. And so one thing that we can do to really help kind of save as many lives as possible is to be able to detect cancer at the earliest stages when it's actually still localized. And some of the statistics, at least from the United States, are that if you look at a lot of these cancers, the 5-year survival rate is incredibly low for cancers such as pancreatic cancer, esophageal cancer, and liver cancer. And so if you look at these metastatic cases, for example, for pancreatic cancer, the 5-year survival rate is only 3%. And also for esophageal cancer, the 5-year survival rate is only 6%. And so again, the greatest thing that we can do to save lives for cancer patients is to be able to detect this at the earliest stages when patients can receive treatments that are going to be more effective and hopefully even curative. So you can imagine if you were to detect cancer at stage 1 when it's still localized, in theory, you'd be able to just surgically remove, for example, the tumor, and hopefully that would be curative for the patient. And so we really want to be able to enable this very early detection of disease. And so this is a question that has really been driving my lab's efforts over the past few years. How can we detect cancer at the earliest stages when we're able to most effectively, again, treat patients that would enable better outcomes for these patients and hopefully better survival as well? And so really thinking about this question of how can we, detect this at the earliest stages, we wanted to understand what is happening at these very earliest stages within these cancer cells. And if you think about, this RNA landscape that I was telling you about, these, 75% of the 3 billion base pairs in the human genome that are being transcribed into RNA, we wanted to address this question of what happens to this vast landscape during cancer initiation. And so we focused on this family of genes called RAS genes, which are mutated in about 30% of all human cancers. And so typically these mutations are gain-of-function mutations that lead to constitutive signaling of this RAS pathway, which leads to hyperproliferation and other changes within the cell. And if you look at some of these cancers, sorry, it's a little bit small on the screen, but Pancreatic cancers are driven by mutations in the KRAS gene, which is part of this RAS gene family. And so the vast majority of pancreatic cancers are actually initiated by this gain of function mutation in KRAS, about 90 to 95% of the time. About half of colorectal cancers have mutations in KRAS. This is thought to be a second hit. But also in lung cancer, this is also thought to be an initial mutation in about a third of lung adenocarcinomas, where this mutation in KRAS is again driving this initiation of cancer. And the kind of open question when we were studying this is, how does this mutation in this very fundamental signaling pathway that's oftentimes perturbed in cancer really affect this non-coding part of the transcriptome? And so here in the pie chart on the bottom right, you can see kind of the composition of the human genome. And so what's really interesting is that over half of these 3 billion base pairs in the human genome are comprised of these repeat elements. For example, these retrotransposons, such as lines, signs, or LTR-derived elements. And so these are, for example, retrotransposons that have inserted throughout the human genome over time. And so it's thought that there are over 5 million of these repeat element insertions scattered throughout the human genome. The great thing, though, about these retrotransposon sequences is that when they are transcribed into RNA, they're actually polyadenylated. And so oftentimes when we generate these libraries that we sequence, for example, on the Nanopore platform, these are all there within the library, but oftentimes people don't really quantify this signal because it gives you kind of these multi-mapping reads. But the nice thing about, again, Oxford Nanopore's platform is that you get full-length information. And so this enables kind of this locus level quantification as well for some of these transcripts. And so we wanted to kind of study this initiation of cancer using these in vitro models. And so what we did in collaboration with my colleague Angela Brooks's lab at UC Santa Cruz, we used these human lung airway epithelial cells that had this mutation in KRAS, this gain of function mutation that's found very recurrently, for example, in lung cancer. And we looked at these cells that had been transformed with this mutant KRAS transgene. And so what's great about this system, this in vitro system, is a very minimal system where you're just looking at cells that either have this mutation in KRAS or a control population of cells that don't have this mutation. And what you can see is that if you implant these cells in a mouse, for example, that they will form tumors if you have this mutation in KRAS, whereas in the control cells, they won't form tumors. And so this is really a very clean system with which to model this initiation of cancer simply by this mutation in KRAS. And so the first thing that we saw was that when you look at the extracellular vesicles, or EVs, that are being secreted from these cells, this is a really simple experiment where you just take the cell culture media and you isolate these EVs. We could see, for example, using nanoparticle tracking analysis, the size distribution of these EVs does change with this mutation in KRAS. And so if you look at the control cells, you see this very nice sharp peak at about 196.5 nanometers in diameter. Whereas when you have this mutation in KRAS, you start to see this heterogeneity in this population of EVs that are being secreted from these lung cells that have this mutation in KRAS. And you also start to see slightly larger vesicles at about 213 nanometers in diameter, as well as smaller vesicles at 150 and 90 nanometers in diameter as well. And so when you take these vesicles and you sequence the RNA, you're able to identify all of the RNAs that are significantly enriched in the mutant KRAS population. And so we saw that a lot of these RNAs were comprised of these retrotransposons, or repeat sequences, that are found again throughout the genome. And so this was really exciting to us because, to our knowledge, no one had really seen that this mutation in KRAS is really turning on all of these repeat elements that are then being preferentially secreted in these extracellular vesicles. And so the model that we favor right now is that when you have this mutation that initiates cancer, for example, in the context of lung cells, what happens is that there is this aberrant upregulation of these repeat element RNAs, for example, these transposable elements or retrotransposons that are found again throughout the genome. And so these are creating these non-coding RNAs that are then preferentially packaged and secreted in these extracellular vesicles that are then sent out into the extracellular matrix. And so we wanted to see, given what we're seeing in vitro in these cell culture models, is this actually something that's happening in patients? Can we detect the same repeat element or this transposable element RNA signal within the blood of patients that have cancer as well? And so we developed this platform, which we call Complete-seq. And so this is an RNA-based liquid biopsy platform, which again enables us to profile all of the RNAs, especially these repeat element-derived transposable element RNAs that are coming from these cancer cells and being secreted into the blood, for example. And so this is a platform where we can take a tube of blood, we can isolate the liquid fraction of the blood, the plasma, and then isolate cell-free RNA. So again, this is the RNA that's being released from these cancer cells and also other cells in the body. And again, this is typically packaged in these extracellular vesicles, so they're protected from degradation. And we can perform RNA sequencing, whether it's long-read RNA sequencing using the Nanopore platform or different short-read platforms as well, where we can then do this repeat-aware analysis. So what this means is that we're quantifying the signal that's coming from all of these different parts of the transcriptome. And so what we're doing is we're quantifying this using a custom annotation of the transcriptome. And so oftentimes people look at the gen code annotation. So these are all the known protein coding genes, but we're also doing something a little bit different. So we're doing what we call a repeat-aware approach. And so what we're doing is we're introducing all of these repeat element sequences. So again, as I mentioned before, there's about 5 million of these repeat elements that are inserted throughout the human genome. And oftentimes when people quantify what's called the transcriptome, they're really referring to either the protein coding genes or this gen code-like annotation of the transcriptome, where you're not getting these repeat elements quantified in your RNA-seq data. And so again, when we take these 5 million sequences and we use that to create this custom transcriptome annotation, we're able to, again, quantify the signal that's coming from these repeat elements. And we're also doing some machine learning modeling to really be able to classify whether someone has cancer or not. And so when we do this modeling, we want to do this feature reduction where we're taking these 5 million repeat-derived features, we're aggregating them down to the subfamily level. So this gives us about 15,000 features instead of these 5 million features. And so we're introducing that as well as the protein coding and other gen code annotated RNAs into our models to train these machine learning models to enable cancer classification. And so what we're able to identify when we do this complete seq-based RNA liquid biopsy is that we can detect about 8 to 10,000 different protein coding RNAs that are in the circulation in the blood. We can see up to about 1,000 or so long non-coding RNAs, and also up to 1,000 or so of these transposable element subfamilies. And so here what I'm showing you is on the left side, these are healthy individuals. So this is healthy blood from these individuals. And then on the right of these plots, this is blood from pancreatic cancer patients. And so, again, we're seeing this difference in the signal of the RNA that's being secreted. Oftentimes, we see more RNA in the pancreatic cancer patient blood than we do in healthy individual blood. And that's true for both gen code protein coding RNAs as well as these long non-coding RNAs. And so when we did this long read, sequencing of these cell-free RNAs, and to our knowledge, this is the first time anyone has really published this work where we're able to do this full-length characterization of these cell-free RNAs. What we're able to see is that there are RNAs up to 1,000 base pairs in length. And there may be even longer RNAs. This might be limited by the processivity of the reverse transcriptase, for example. But to our knowledge, again, this is the first time people have been able to show that there is this true full-length cell-free RNA within these patient blood samples. And the dogma in the field had been that, all of this RNA is just going to be degraded and it's not going to be full length, and you're not going to see, signal coming from, for example, fully spliced messenger RNA or other types of RNA. But when we did the experiment with the Nanopore platform, it really enabled us to get the true identity of these cell-free RNAs, the true length of these RNAs. And so here what I'm going to show you is just one example of a protein coding gene that's transcribed and secreted into the blood of pancreatic cancer patients. So here what I'm showing you is a UCSC genome browser plot. So this is just a screenshot of what you would typically see in the genome browser. But here you can see on the bottom right, this is a protein coding gene, PLAT4, that is significantly upregulated only in pancreatic cancer patient blood samples. And so if you're not familiar with looking at these annotations of these genes, what I'm showing you here in these fatter bars, these are the exons, and then here in these little thin bars, these are the intronic sequence. And so anytime you see these black lines, this indicates a single molecule that's been sequenced on the Nanopore platform that is a cell-free RNA that's circulating within these patients' samples. And so here you can see that this RNA is fully spliced. So you can see the exonic sequences And it's also full length. So you're seeing a full length messenger RNA that corresponds to this protein coding gene that's been secreted into the blood of these pancreatic cancer patients. But what's really quite interesting, and which again is only enabled by this long read technology, is when we look at these patients, we also see these novel RNA isoforms that are not in the annotation of the transcriptome of the human genome. And so here what we're seeing is that there are these RNAs that are being transcribed more than 50 kilobases upstream of the known transcription start site of this protein coding gene platform. And so you can see that there are exonic sequences, again, more than 50 kb upstream of the transcription start site here. And these are novel RNA isoforms that we were able to discover only in cancer patients and not in healthy individuals. And so this is really a powerful platform and powerful technology that really enables us to identify RNA signals that are completely specific to the disease and again not found in healthy individuals, which gives us these very nice biomarkers that are cancer specific. And also, again, going back to these repeat elements, we see that there are a lot of these repeat elements that are also transcribed. The really interesting thing that we saw was that if you look at these sign-derived cell-free RNAs, so a full-length typical sign element in the human genome, which is an ALU sequence, is about 300 base pairs in length. So we see that a lot of these are exactly this 300 base pairs in length. The other thing that I'll mention about these ALU sequences is that these are typically primate specific. Actually, they're exclusively primate specific. And so there's about 1 million of these ALU insertions in the human genome. And so if we were to model, for example, this process in mice or study the blood of mice models of cancer, we would never see the signal because these ALU primate-specific transposons are not found in the mouse genome. And so really we're getting this very nice, human and primate-specific signal that's being turned on in the context of cancer. And if you look at, for example, all of the RNAs that are significantly upregulated in pancreatic cancer, so here in the volcano plot on the right, I'm showing you all of the cell-free RNAs that are significantly enriched in the blood of pancreatic cancer patients versus on the left, these are all the RNAs that are enriched in healthy individuals. And so you can see in this circled area that the strongest signal in terms of the fold change that's being secreted into the blood of these pancreatic cancer patients is coming from these ALU sequences, these primate-specific elements that are being aberrantly turned on and transcribed and secreted into the blood of these patients. And so we're really excited about this observation because it enables us to identify signals that are specifically enriched in the disease context. And so we also wanted to see, is this more generalizable to other types of cancers? So as I showed you, the pancreatic cancer data, when we looked at other types of cancer, so we've looked at about a half dozen cancer patient samples, looking at their blood so far. Here I'm just going to show you 2 representative examples. So if you look at liver cancer patients, on the right of this plot, again, these are all of the cell-free RNAs that are enriched in the blood of liver cancer patients, versus on the left, that's a signal coming from the healthy individuals. And you can see there's a lot of retrotransposon signals. So any dot that's colored is a non-coding RNA that's coming from these repeat elements. And you can also see, for example, in esophageal cancer as well, there are these cancer-specific RNAs that are being turned on. It's not as pronounced as in liver cancer, but the signal is very different. And so what we're able to see is that there are these tissue and cancer-specific signals that are coming from these repeat elements as well. And so the great thing about this complete seq-based approach is that it enables us to both quantify these repeat element RNAs, but also use them as additional features within our machine learning models to be able to identify whether someone has cancer or not. So here what I'm showing you is a model that we trained using what we call a repeat naive approach versus a repeat aware approach. So here in the orange color, this is a repeat naive approach where we're training this model only using the GenCode derived cell-free RNAs versus the model that's trained using these repeat element features, this repeat aware model in black. And so you can see that the performance of these models is significantly better in the repeat aware approach, where we're getting much higher sensitivities at the same level of specificity for liver cancer. And then also for esophageal cancer as well, we see a significant improvement in the performance of these machine learning models that enables us to classify whether someone has esophageal cancer or not. But given the performance of the esophageal cancer model is not as good as, for example, the liver model or some of the other models that we've trained, we wanted to do a deeper dive into esophageal cancer to see if we could do better in terms of being able to identify these cancers. And so to do this, we collaborated with one of the world kind of leading labs in this space, which I'll talk about in a second. But the reason why we wanted to focus on esophageal cancer initially is because we know that, for example, in the United States, 1 in 5 adults suffer from gastroesophageal reflux disease. And so when you have reflux disease, this predisposes you to this pre-malignant condition called Barrett's esophagus, where if you leave it unchecked, this can lead to increasing dysplasia and the formation of esophageal adenocarcinoma. And so, as if you recall from the statistics that I showed you earlier, for patients who detect esophageal cancer at very late stages, when it's already started to spread and metastasize, the 5-year survival rate is only 6%. And so, even though the likely risk of getting this cancer is relatively low, if you do get this cancer, it's extremely problematic in terms of being able to treat it effectively. And so we wanted to see, can we detect this cancer at very early stages? And can we even detect cancer or precancer, actually, when patients have Barrett's esophagus with increasing dysplasia? Could we be able to identify these patients who are at higher risk for developing esophageal cancer and potentially even treat these individuals to prevent cancer as well? And so to do this, as I mentioned, again, we collaborated with Rebecca Fitzgerald's lab at the University of Cambridge, where they have this really great study called the OCCAM study, where they're looking at individuals who have Barrett's esophagus or esophageal cancer, and they've looked at about 3,000 or so individuals in the UK that have these different conditions, and they've isolated, for example, blood samples from these individuals. And so they kindly sent us a small cohort of blood samples from individuals who had either esophageal adenocarcinoma or high-grade dysplasia in Barrett's esophagus. And so what we did was we, again, used our complete-seq platform to identify all of the cell-free RNAs that were being secreted in the blood of these individuals who either had high-grade dysplasia or esophageal adenocarcinoma at early stages, and compare that to a control cohort of individuals who didn't have this cancer or dysplasia. And so again, what we're doing is we're doing RNA sequencing in the cell-free RNA that's secreted from these patient tumor cells. And then we're doing, again, a repeat aware analysis to be able to enable disease classification. And we did this in collaboration with my UCSC colleague, Karen Miga's lab, to do this Promethean-based full-length sequencing of these cell-free RNAs. And so really the most surprising observation from these studies is that we were able to discover almost 287,000 novel unannotated cell-free RNA transcripts that were in the blood of these individuals that had either high-grade dysplasia or esophageal cancer. And so really this is enabled, again, by this long-read platform. And so if we were to do this with short-read sequencing technology, we wouldn't have been able to identify these novel unannotated transcripts. But because we're doing, again, this full-length sequencing, we see the true nature of these transcripts within the human genome. And so we're really excited about this observation because it's really giving us much greater signal to be able to do early detection of disease or even detection of precancer as well. And so here what we're seeing is that for these protein coding genes, again, we see about 10,000 or so detected in healthy individuals or individuals with a cancer or Barrett's with high-grade dysplasia, and also for these other biotypes that are in the GenCode annotation. But again, when you look at these novel unannotated transcripts, we see that there are tens of thousands of these unannotated novel cell-free RNAs in any given individual. And so this is really exciting for us to be able to enable early detection of disease. So here, just to give you a sense of what this full-length cell-free RNA looks like, we can see that in the control healthy individuals versus the cancer patient samples, that we could see cell-free RNAs of up to almost 1,000 base pairs in length, as we saw in the pancreatic cancer study. One thing that I do want to say as a caveat is that this may not be the true biological distribution of the cell-free RNAs. So here what we're doing is a cDNA synthesis first before we do the long-read sequencing. And so due to the nature of the reverse transcription, if it's not processive and if it's not giving us longer cDNA molecules, this could be also reflecting some of the technical limitations of just the reverse transcription. And so as Ellie mentioned, it would be great to be able to do direct RNA sequencing in the future. But currently, the input requirements for direct RNA are relatively high, and the amount of cell-free RNA that you can get from blood is relatively low. So there are some technical challenges in the present time. But regardless of that, when we look at the cDNA, we see that there is a difference in the size distribution of healthy individuals in their cell-free RNA as well as these cancer patients. And so when you do some statistical tests to identify whether there are significant differences between these control distributions and these cancer patient distributions, we do see that there is a significant shift towards these shorter cell-free RNA molecules within the blood of these cancer patients. We're not really sure what the biological mechanism for this may be, but this is also, again, an interesting feature within these cell-free RNAs. And so, as you heard, there are these efforts to do DNA-based fragmentomics, and so maybe RNA-based fragmentomics or these size comparisons may also be something that we could pursue in the future. But regardless of that, when we looked at the differentially expressed RNAs, again, on the right, these are all of the RNAs that are enriched in the esophageal cancer patients versus on the left, these are the healthy individuals. We can see that there are certain types of genes that are enriched. For example, these mitochondria-related genes that are being aberrantly transcribed and secreted into the blood in these esophageal cancer patients. And so we wanted to kind of look at this using a much larger cohort. So what we did was we looked at all of the RNA sequencing data that's available, for example, in the Cancer Genome Atlas. This is an effort that the US NIH has funded, where they've looked at over 33 different types of cancers across over 11,000 different patients. And we took the RNA-seq data from these 11,000 different patients and compared it to, for example, healthy tissue RNA-seq data. So this is also NIH-funded efforts by the GTEx consortium to look at about 50 or so different healthy tissue types and to do, again, RNA sequencing. And so in aggregate, we're looking at about 26,000 different RNA sequencing samples across healthy individuals and cancer patients to identify the signal that's specific to esophageal cancer. And so here, what I'm showing again is a volcano plot where we're doing a one versus all comparison of just the esophageal cancer patients versus everything else. And so we can see that there's a lot of signal that is specific to the esophageal cancer tissue and tumor. And also in green, these are the repeat-derived signals. And so what we're really excited about is that when we're doing these tissue-specific profiling experiments and we're quantifying all of these libraries using our repeat-aware approach, now we're able to deconvolve the signal to be able to say these RNAs are coming from these specific tissue types or these specific disease types. And so if you were to do this sequencing-based approach, this liquid biopsy approach, for any individual, even if we didn't know what cancer they had, if we saw specific cell-free RNAs, we'd be able to say that this is most likely coming from this particular tissue. And so this would enable a follow-up through various imaging modalities to confirm whether there is a tumor, for example, in the esophagus or in the liver or the lung or other tissues. And so just to give you a sense of what this esophageal cancer cell-free RNA data look like, so here what I'm showing you on the top is the annotated long non-coding RNA CITOR's kind of genome annotation for their different transcripts. And so what you can see is that there are 5 different isoforms that are characterized in the human genome. And if you look at the cell-free RNA from esophageal cancer, again, this is full-length, cDNA coming from these blood samples. You can see that there are multiple isoforms of this cell-free RNA that are present in a single individual. And so here you can see through the long-read data that, for example, there are cell-free RNAs that have this isoform, whereas there's others that don't. There are some that are this shorter isoform as well. So we're really able to kind of understand the true complexity of the cell-free RNA transcriptome through this long-read technology, again, that enables us to look at the true length and true isoform nature of these cell-free RNAs. And again, the great thing about our platform is that we can also look at the repeat element signal and quantify that as well. So here what I'm showing you is a protein coding gene on the top right, and all of the, again, RNAs that, cell-free RNAs that align to that particular protein coding gene. But what you'll notice is that there are a lot of these kind of shorter blocks of black you can see coming from, for example, these sign elements that are just present throughout these cancer patients. And so again, there's a lot of signal coming from both the protein coding part of the transcriptome, but also these repeat elements as well, which we're, again, leveraging to do these machine learning model-based approaches to be able to classify disease. And so here, this is just another example where you see different isoforms, and you can start to appreciate some of the complexity of these cell-free RNA transcriptomes. And then here, again, you're seeing kind of this aberrant upstream transcription of these cell-free RNAs. So here, again, on the top is the annotated RNA from this particular gene. And then here, these are these novel annotations, including these upstream 5' upstream transcription start site RNAs that are, again, kind of found within these hotspots of transposable element transcription as well. And so just one last example here. This is an example, again, of a protein coding RNA where, again, in black, you see all of these cell-free RNAs that are in the circulation. And then a little bit upstream of that, we're seeing this novel RNA, again, part of these 269,000 novel unannotated transcripts in the human genome. And so here you can see that for this particular cell-free RNA, it's actually comprised of multiple repeat element sequences, both SINE and LTR-derived sequences. And so, again, this really enables this very cancer-specific signal that we can use, again, to identify whether someone has cancer or not. And so just to kind of summarize what I've been telling you about so far, we're really excited about the future of nanopore-based RNA liquid biopsy technology because, first of all, it really enables a systemic understanding of disease in vivo. So as I mentioned in my earlier slides, really the signal is coming from every tissue and organ system in the human body. And so there's really no other way to get this systemic understanding of disease in vivo because to do anything even comparable to what RNA-based liquid biopsies enable, you'd have to do a surgical removal and a tissue biopsy of every tissue within our bodies. And again, that would only capture a fraction of the heterogeneity because even like any given organ or tissue is very heterogeneous. And even for cancer as well, if you were to do just a small biopsy of a tumor, you'd know that that part of that tumor is most likely very different from other parts of the tumor. And so really this kind of non-invasive liquid biopsy-based approach is really enabling us to get signal from every part of the body, every part of the tumor, and really giving us this very comprehensive picture of what's happening at very early stages of disease. And then the other thing that we're really excited about is that this platform enables us to do this novel cell-free RNA biomarker discovery. So as I mentioned, over 269,000 novel unannotated cell-free RNAs that, again, is enabled by this long-read technology, where we're able to use this information to determine whether someone has cancer or not. And so we've done some different studies, some machine learning based modeling where we're using these features. And so we see extremely high sensitivity, even at very early stages of disease, stage 1, even at precancerous stages as well. So we're excited about the ability to both detect cancer at the earliest stages, but also to detect these precancerous conditions as well. And then lastly, we're hoping to be able to develop this for example, the MinION platform. I know this is the older version of the MinION, so I should update it. But this really handheld approach where we can take this to remote and resource limited settings with just a handheld sequencer and a laptop. We really want to enable a very kind of cost-effective and accessible and equitable diagnostics using this RNA-based liquid biopsy platform. And so we've been trying this, for example, with the Flongle flow cell, and so we're starting to see some promising results there as well. So with that, I just want to thank all the amazing people that contributed to this work. So the Complete-Seq technology was really initiated by a former PhD student in my lab, Roman Reggiardo, who's now doing his postdoc at Stanford in Howard Chang's lab. And also the Nanopore work was really initiated by a current PhD student in the lab, Vikas Padu, who has really pioneered a lot of this Nanopore technology development, as well as the computational analyses to identify these novel isoforms and leverage them for diagnosing disease. Also, the other people in the lab that have really contributed to developing this platform and technology include Suri, who's just finished her PhD, Andy, who did all of the GTEx and TCGA information, Alex, who's been doing a lot of the short read work and combining it with the long read technology as well, Connor, who's also been helping us with the development of this technology as well, and just all the other amazing people in the lab that have contributed to this. Especially want to thank my collaborators, Angela Brooks and Karen Miga, and also Dave Deemer at UCSC, who's really been invaluable in terms of helping us to really just do more of this Nanopore-based sequencing. Also, Rebecca Fitzgerald's lab at Cambridge, our colleague, former UCSC colleague, Matane Jane, who's now at Northeastern, and also my other collaborators as well. Especially want to thank the American Cancer Society, which has funded the vast majority of this Nanopore sequencing-based work for really enabling us to do this very high-risk research, but we think that this is really going to be transformative for early detection of disease. And so with that, just want to thank you all for joining. Thank you for your attention.